Hi students, now let us going to see the subject biochemistry. The topic is lipid metabolism. In the lipid metabolism, now we are going to discuss about the beta oxidation. Okay, so beta oxidation means oxidation of long chain fatty acids converted into acetyl coenzyme A. Okay, long chain breakdown of the long chain fatty acid into acetyl coenzyme A. Okay, this is the beta oxidation process. Okay, in the beta oxidation of the fatty acid, it is involved three stages. Okay, three stages are available here. The first one is the activation of the fatty acids. Okay, fatty acid activation is happening in the cytosol. Okay, cytoplasm area. And the second one, transport of the fatty acid into mitochondria. Okay, in the cytosol, it is activated. The fatty acids are activated. Then it is transported into the mitochondria. Okay, and the third step, beta oxidation proper in the mitochondrial matrix. Okay, this is the mitochondrial matrix. Okay, so inside the beta oxidation proper, okay, happening. Okay, these are the three stages of the beta oxidation. Okay, fatty acid activation and transport of the fatty acid and beta oxidation proper. Okay, let us see one by one. The first one, fatty acid activation. Okay, fatty acids are activated, then it is converted into acyl coenzyme A. Okay, by the enzyme, by the help of the enzyme, the name of the enzyme is thiokinases or acyl coenzyme A synthetases. Okay, so with the help of the thiokinases or acyl coenzyme A synthetases, the fatty acids are converted into acyl coenzyme A. Okay, so during this activation period, it requires, okay, for fatty acids activation, it requires ATP energy. Okay, and the coenzyme A and magnesium ions are also required. Okay, now the fatty acids react with the ATP to form the acyl adenylate. Okay, so then it will be combined with the coenzyme A to produce the acyl coenzyme A. Okay, here in this activation period, two high energy phosphates are utilized. Okay, so that's why the ATP is converted into pyrophosphate. Two energy phosphate molecules are required or utilized here. Okay, it is converted into pyrophosphate. Okay, so here the enzyme is involved. The name of the enzyme is inorganic pyrophosphatase. Okay, it hydrolyzes pyrophosphate into phosphate. Okay, this enzyme hydrolyzes the pyrophosphate into phosphate. Second step, transport of acyl coenzyme A into mitochondria. Acyl group of the acyl coenzyme A is transferred to the carnitine. Okay, so acyl coenzyme A is transferred to the carnitine. Okay, by the enzyme activity. Okay, what are the enzymes here involved means carnitine acyltransferase 1. Okay, the name of the enzyme is carnitine acyltransferase 1 enzyme. This enzyme is found in the outer surface of the inner mitochondrial membrane. Okay, this is the mitochondria. It contains outer membrane and the inner membrane. Okay, so this is the outer membrane, this is the inner membrane. Okay, this enzyme is found in the outer surface of the inner mitochondrial membrane. This is the area. In this location, this type of the carnitine acyltransferase enzyme 1 is found. Okay, so this enzyme is involved in the reaction of the acyl coenzyme A is transferred to the carnitine. Carnitine means beta-hydroxy gamma trimethyl aminobutyrate. Okay. So then now the next step, the carnitine acyltransferase 2 enzyme is also involved. This enzyme is found in the inner surface of the inner mitochondrial membrane. Okay, here this is the inner membrane, this is the inner surface. Inner surface of the inner mitochondrial membrane, the second enzyme, that is the carnitine acyltransferase 2 enzyme is found. Okay, what is the role of the second enzyme? It converts the acyl carnitine to acyl coenzyme coenzyme A. Okay, it converts the acyl-carnitine to acyl coenzyme A. Now the carnitine, okay, it released written to the cytosol. Okay, again it is written into the cytoplasm. Okay, the first reaction activation of the fatty acid is done in the cytoplasm. Then it is transferred into the mitochondria. Then again it is written into the cytosol for the reuse. Okay, so usually the cell has two separate poles. Okay, one is the cytosol and another one is the mitochondria. Okay, two places are here. Okay, the cytoplasm and the mitochondria. Okay, so and the third one is the beta oxidation proper. Okay, beta oxidation proper, here 4 steps are following. The first one is the oxidation, and the second one is second step also hydration, third step also oxidation, and fourth one is the cleavage. 4 steps are available here. The first one is the oxidation. Okay, oxidation, you know, addition of the oxygen or removal of the hydrogen, isn't it? Okay, oxidation reaction. Here what is the reaction is going on? Acyl coenzyme A undergoes dehydrogenation by an FAD-dependent flavoenzyme, acyl coenzyme A dehydrogenase. Okay, here the product is the acyl coenzyme A. Okay, it is converted. Okay, it undergoes the dehydrogenation. Dehydrogenation means the removal of the hydrogen. Removal of the hydrogen. Okay, so the role of the enzyme. What is the name of the enzyme here? Acyl coenzyme A dehydrogenase. Okay, this is the particular enzyme. This is the FAD-dependent flavoenzyme. Okay, this acyl coenzyme A dehydrogenase is a FAD-dependent flavoenzyme. Okay, in this area the double bond is formed between alpha and beta carbons. Okay, and the second step is the hydration. Hydration means Enoyl coenzyme A hydratase brings about the hydration of the double bond to the form. Okay, then hydroxy acyl coenzyme A. Okay, Enoyl coenzyme A hydrolase brings about the hydration. Okay, hydration, it is converted into hydroxy acyl coenzyme A. And the third step oxidation again. Then beta hydroxyacyl coenzyme A. Okay, dehydrogenase. Okay, this enzyme catalyzes the second oxidation. Again oxidation reaction is happening here. At the time the product is beta ketoacyl coenzyme A. Okay, this is the third step. In the third step the product is developed, that is beta ketoacyl coenzyme A. Here the enzyme, the name of the enzyme, the involvement of the enzyme is beta hydroxyacyl coenzyme A dehydrogenase. Okay, then the fourth step, the final reaction in the beta oxidation is the liberation of the 2 carbon fragment. What are the 2 carbon fragments? Acetyl coenzyme A from acyl coenzyme A. Okay, acyl coenzyme A is obtained from the acetyl coenzyme A. Okay, so by the enzyme, the name of the enzyme is beta ketoacyl coenzyme A thiolase. Okay, this enzyme is involved in the final reaction of the beta oxidation. Okay, these are the steps of the beta oxidation proper. Okay, so in the beta oxidation mean metabolism, it is one of the lipid metabolism. 3 steps are here. First step is the activation of the fatty acid. Fatty acid activation. Okay, how the fatty acid is activated. Okay, this fatty acid activation may be done in the cytosol. And the second step is the transport of the acyl coenzyme A. Okay, this acyl coenzyme A transport may be done in the mitochondria. Okay, inner mitochondria and outer mitochondria. And the third step is the beta oxidation proper. Here 4 steps are followed: the oxidation, hydration, oxidation and cleavage